one last night. I think you still can check it out on YouTube. So before we introduce Professor Likens, I just want to make an announcement that there's no department seminar next Friday. And 2 weeks from now, it will be Dr. Kristen Faria from Woods Hole Oceanography Institution. She'll be talking about submarine volcanic eruption. So if you are interested in submarine volcano, stay tuned. Yes.
But next week we will have Rick Zimmerman in this room from 2 to 3. You can come back. Who will be eager to talk with students about opportunities in industry. He is president of an oil and gas company in Houston, and so I encourage all students and maybe some faculty that are looking to change their job.
Postdocs.
Postdocs, yes. Anybody that wishes to should encourage undergraduates as well. So.
And graduate students at this regular seminar time, we're going to have our graduate student meeting.
At 3.
At 3, yep.
If anybody stays.
It's like a perfect jigsaw. So I guess you will hear more about it next week. So again, stay tuned. So today we will have Shujay to introduce our speaker.
Thanks, Monghan, and thanks for coming. And some of you were at the health lecture, where we talked about Gene and some of his achievements, being a member of the National Academy of Sciences, winning the National Medal of Science, the highest honor in the United States. But I wanted to talk a little preface about his talk and some long-term observations of Gene himself. So,
Do it without your phone.
okay. So, as geologists in the earth sciences, we're basically looking at signals from the earth. We may be looking at neutrinos from deep in the earth and looking at making sense of those patterns. Or we may be looking at isotope records in a rock and looking at changes over time to infer some kind of pattern. Or a tree ring, looking at isotopes. So an isotope record. You can look at the chronology or what happens in terms of changes in running waters and streams and rivers, but it's not, we can't get a core. Sometimes we can't, we can't look in one location for a record. The way to do that is basically you go out with a bottle and you sample a stream repeatedly over and over and over and over again for decades. And that's what Gene and his collaborators have done. Basically, they've pieced together the longest continuous and most detailed record of stream chemistry in North America, maybe the world, looking at different analytes, and they've been able to track changes in processes in the Earth's surface that are related to acid rain, deforestation, and other environmental changes like climate change. Now, that's something just relating what Gene does to what we do in our department, changes over time and inferences. I wanted to also mention that when you look at Gene's data, it has an application to everyone in this room, particularly students. And Gene himself is a student. When we were eating at Board and Brews the other day, Gene was talking about, he was saying how this reminded him of Madison, Wisconsin, where he went to graduate school, and his eyes lit up. And even when I picked him up at the airport, he was talking about being a graduate student. No matter what award Gene has won, when you talk to him, he's like a graduate student. I mean, if you really know him, he still eats pizza. You can take the kid out of graduate school, but you can't take graduate school out of the kid. If there's free food, I see Gene going for it. But, seriously, he still has a joy for science and figuring things out and having fun. And for you students out there, I don't know what's going to happen in your life, what direction it's going to take, but I do know one thing about your life. you're going to have some ups and downs in your life. You're going to have bad years, maybe bad decades, good years, and good decades. When you look at gene stream chemistry records, okay, that span 5 decades, you're dealing with changes in analytical techniques, changes in funding climates, changes in climate, changes in people, okay? People are the hardest aspects of science to deal with, okay? Gene had to deal with a lot of collaborators and different people. So you're dealing with a lot of changes. Gene kept going. He kept going and going. He's still here. He gave 2 talks. So whatever you do in life, you know, as Gene went on, he had to come up with new questions. NSF isn't going to fund a question from 50 years ago, right? You have to come up with new questions. Gene's always constantly reinventing and recasting his approach, whether it's his science or things that happen in life. And so when you guys go through life, I think, and you look at Gene's body of work and what he's really done, just think about this. You have to, you know, stay curious, keep asking questions, and then also have fun, and the most important thing is to just keep moving. And he's still moving at 80, 83, 84, and he's still going strong. Sorry, Gene, I didn't make that.
Is that 38? Thank you, Sujay, for that really kind introduction, and thank you. I see many faces that were there yesterday. Thank you for coming back. I have a couple caveats that I'd like to start today with. First of all, I'm going to have some slides in the talk today that I had yesterday. I didn't know how many of you might be new today, and so I want to make one point, and so I need to show some slides from yesterday to make that point. I don't like showing the same slides in subsequent lectures, something I really try not to do, but for those of you that were there last night, there are a few that are the same. There's a section on acid rain that's the same. Secondly, what I'm going to talk, I'm a biologist, and so what I'm going to talk about today is what I've spent most of my life doing research at the Hubbard Brook Experimental Forest, trying to understand how complicated ecosystems function. We recently, just a year ago, put all this together in a book, Dick Holmes and I. It's called Hubbard Brook, The Story of a Forest Ecosystem, published by Yale University Press, and that wasn't an advertisement, but this talk is from that book. It's our attempt to bring together, synthesize what we've done for 50 years. Actually, it's been 55 years, 56 full years. Yeah, I know some of you weren't even born then, but that's how long we've been trying to understand how the system works, and it really takes that long. And finally, I want to thank the National Science Foundation, who has funded this research from the beginning. I've had continuous support from the National Science Foundation, maybe some kind of a record, I don't know, I don't care, but it's been really critical to do the research with that funding, and particularly with the more controversial acid rain work. I could always say that, you know, I'm not being funded by this group or this industry, that industry. I was funded by the National Science Foundation, where I had to compete for those funds, very tight funding throughout all that period. Some years we had to do a proposal every year, some years every other year. I remember turning in a proposal literally 215 pages, single space, and the main criticism was it wasn't comprehensive enough. So that long effort I'm actually very proud of, but I wanted to acknowledge that. Okay, so Hubbard Brook is located in the White Mountains of New Hampshire in the northern hardwood forest biome. It's a large biome. It's characterized by yellow birch, sugar maple, beech, American beech trees, some hemlock, and a balsam fir, red spruce. It's that kind of a northern hardwoods system. It's a mixture of coniferous and deciduous trees. Right now it's leaf peeping season. The colors are gorgeous. So people are coming to this part of New England, of the world, from all over to look at the beautiful colors. That is one of the characteristics of this forest. The Hubbard Brook system is a large bowl-shaped valley that has headwater streams, has a lake at the bottom of the valley. It is organized into watersheds, and we call these watersheds watershed ecosystems, a hyphenated word, because my research vessel is this unit. Yours might be a flask on the desk in your laboratory, might be something else. This is my unit, and the unit is characterized by boundaries, so that there are lateral boundaries that define where the water flows. Those are called phreatic divides. And then the system, fortunately, has granite underlying the unit, so that there is no deep water loss. That's critically important, because if there were deep water loss, we couldn't measure it quantitatively, either its chemical content or its amount. This is not like that. So in this case, a gauging weir is physically attached to the bedrock. We found some place where we could physically attach the weir to the bedrock. And then all the water that's moving downhill is forced to pass through that gauging weir, so we can measure it quantitatively year-round. In the wintertime, the ponding basin behind the V-notch weir is heated so that the ice doesn't build up in the V-notch, and so we can get quantitative measurements year-round. We have that unit then, which we measure all the incoming precipitation and all the outgoing stream water, and that gives us a hydrologic balance, and then knowing the chemistry of both of those components, we can do chemical balances at the same time. The large bowl-shaped valley is about 6,500 acres in size, 3,200 hectares in size, roughly. They keep adding a little bit of land, so it changes. It's drained by a fifth-order river, but in New England we call rivers all kinds of names, and so it's Hubbard Brook. It's not a brook, it's a big river. And adjacent to, didn't want to do that. Adjacent to the Hubbard Brook here, which drains the whole valley and provides the name, are a series of smaller streams which we have positioned these gauging weirs, and those are called experimental watersheds. This is the Hubbard Brook Experimental Forest. And so these south-facing watersheds, 1, 2, 3, 4, 5, 6 of them, have been used most extensively. There are also 3 north-facing watersheds. These have more coniferous vegetation, these have more deciduous vegetation, and then there's one here that has not been used experimentally. So that's the whole system. Mirror Lake is at the bottom of the valley. We have cored the sediments of Mirror Lake, 12.4 meters of core, a 3-inch diameter core, which is an amazing resource, because with that and dated and analyzed completely, and I mean as completely as we possibly can, chemically, biologically, strategically, we've been able then to understand better what the history of this little unit of land is like, and what happened since glaciation. The glacier melted about 14,000 years ago. It left a block of ice, so this is a lake, a kettle lake, which was formed when that block of ice melted. It provided the water to fill the hole, and we had Mirror Lake, which is the name of the lake at the bottom of the valley. So it's a whole unit. And the whole effort has been to try to understand as best we could air, land, water, interactions, and then how the biology fits in with all that. And from an ecosystem point of view, how all that may fit together and how it may fit together. So there are, I wrote this down because I knew I wouldn't remember it, 258 species of vascular plants, 27 species of trees, 127 species of birds, 100,000 species of microarthropods, 15 species of chemicals that are of major interest to us, 6 species of salamanders, on and on and on. Those are just the species. And then there are the individuals that are within each of those species. So what turns me on, and my colleagues, is how does all of that fit together, and all that complexity, how can I begin to understand it? How can I get my head around it? That's what I enjoy doing. So we started with a very simple metaphor. Could we think of this system sort of like a physician does with a human patient? If you analyze the chemistry of blood or urine, you have some idea of the health of the patient. And then if that chemistry changes in some way, you have to go into the pancreas or the liver or the lungs and say, well, that's why this chemistry changed. Well, if the chemistry of the stream draining my watershed ecosystem changed, then I have to go inside and look at the trees or the soil or the microbes or the whatevers and try to understand why that chemistry changed. That simple metaphor, that medical metaphor, has really served us very well because we use it. And we use it in the way of trying to understand how we can get our head around, our mind around all of that complexity. I know all of you in this room work on complex subjects. All of you do, probably more complex than this. But this is all I can handle. This is enormously complex. And trying to understand how it all fits together is what I love to do and is what I do. So the simple conceptual diagram shown here on the right is our first paper. The first paper we published from this study when we started. The study started in 1963, published the first paper in 1967 in Science. And the paper had this diagram. It wasn't quite this pretty because this is the diagram from the book, which has been prettied up. But it was how we thought the system might fit together in a very simple way. And we've used this diagram, this conceptual diagram. I'm going to preach just for a moment. If you don't have a conceptual diagram, when you start a research project, I think you're missing something. Because a conceptual diagram helps you get your mind around the project, helps you not forget critical things you need to study, helps you be able to put the pieces together. This diagram has served me and my colleagues admirably well for now over 50 years because it does that for us. I'm done preaching. Well, maybe not. So at the base of each of these watershed ecosystems, a weir like this is established. Some of you will know about these. This particular weir has a sandemus flume and a V-notch. This is a 90-degree V-notch, a ponding basin behind it. The height of water in the ponding basin is recorded in a stage height basin here, and then all that gives us the flow. It's calibrated for the height of water, for the flow, and we have a rating curve, and so we can get the flow year-round. As I said, this is all covered over in the wintertime, all of this, and heated, so that we can do it. Because if ice were to form in this V-notch here, then that would mess up our measurements, okay? Likewise, areas are cleared in the forest where we measure the amount and the chemistry of precipitation. This is examples of those summer and winter. Those of you that were there yesterday saw my funnel. Well, there's the funnel, but there's more sophisticated ways we measure amount and chemistry of precipitation. The long-term data that we've been able to acquire from this study, to me, has been remarkable. This is the nitrate concentration in precipitation and stream water. When we first started our study, the stream water nitrate concentration was that. We didn't know what that really meant, but if you look for now 63 through about 1968 or 69, it went steeply up. So we wrote a paper. Oh, look, the nitrate concentration in stream water is increasing. Well, little did we know, that didn't have any relation to the long-term pattern at all. This is now a pattern that goes through 2015. And yeah, it went up, but then it went down and jig-jagged all around. And now it's currently dropped to values lower than it was when we started, and approximately what we think pre-industrial revolution values should have been. So it's dropped very low, and I don't know where it's going to go, maybe even lower than that, primarily because of air regulations, which I'm going to talk about in a moment. Stream water had this period. Every one of these years goes up and down. During the growing season, the nitrate concentration is drawn down because the biology takes the nitrate as an important nutrient. And then in the wintertime, the stream concentration goes up because the biology goes dormant, except for the few coniferous trees. So every year, there's that pattern. It's sort of like the famous Keeling CO2 curve. But within that annual pattern, there are these periods in which it's low, then relatively high, then low, and now exceptionally low. It's barely detectable, and we don't know why. We don't know why it's barely detectable, because from the theoretical considerations of how the system works, it shouldn't be that low. And we can come back to that in a moment. Okay. So these are a couple graphs of calcium and magnesium. The calcium concentration shown in blue and the magnesium in green dropped steeply from the beginning of our study until about 1975 or 1980 or so, and then has been relatively flat, particularly in precipitation, less so in stream water, but relatively flat. Again, we don't know why exactly, except during this period, there was a lot of both calcium and magnesium that was coming into the system from air pollution. The 1970 Clean Air Act helped reduce that part of air pollution. So even though this is flowing through this complicated system, we still see that signal in our chemistry, our metaphor. Something is changing in the stream water chemistry. Another important part of this is that we set up what many of us worked very hard to set up national networks for monitoring precipitation chemistry, NADP, National Acid Deposition Program. Some of you may be familiar with that, maybe not. But we now have stations all over the country measuring precipitation chemistry. They started here, 1978. Most of this action was over. We missed in that network. Doesn't mean that network isn't important, it's critically important, but it missed that, it missed that first key part. And wouldn't it be great to know what happened back here? So we've now tried to hindcast our numbers. We're greedy. 55 years is wonderful. but we went longer, so we've now hindcasted back to 1900 to see what may have happened and to frame what's going on currently. Okay. Another major part of the work at Hubbard Brook is that because these watershed ecosystems are my research vessel, I can do experiments in them. And so we experimentally manipulated whole watershed ecosystems. We cut down all the forest, or we did various things. I'm going to talk about that in a moment. That idea of experimentally natural systems started at the University of Wisconsin, started with Chancey Jude, who was doing that with lakes. He was trying to make lakes more productive, to try to get more fish to grow. I was his student. No, I'm sorry, it wasn't his student. His student was Arthur Hassler, and I was Arthur Hassler's student. So I was brought up in that mode of experimentally manipulating large natural systems. The experimental lakes area, which is very famous in Canada, one of Hassler's students was Waldo Johnson, and he started that area. So that's the legacy that produced these large experimental manipulations. So the first one we did was we experimentally cut down all the forest in watershed number 2. It was an experiment. It wasn't a harvest. We didn't remove the trees. We cut by hand all the trees and left them in place. So this is the way it looked after it had been cut. It was cut in 1965. We expected that the amount of water running out of the system would increase as a result of cutting all the trees, because trees evaporate and transpire a lot of water. At Hubbard Brook, the annual amount of precipitation is about 56 centimeters a year. 65% of that runs off in stream water. 35% goes off as evapotranspiration. Of evapotranspiration, T, that's about 75%. So it's a huge water loss that happens just through the trees. Okay? So we expected more water. What we didn't expect was the chemistry of that water changed dramatically. The system was cut in 1965-66, in the winter of 1965-66. The red line shows the nitrate concentration the following summer, starting in June, July, increasing dramatically. Incidentally, this is a slide that I made back when we used to take photographs of actual drawings and take the negative, and with a felt marking pen, we'd color in the lines on the negative and then mount those and show them in talks like. It took me longer to make a digital version of that old slide for this talk than it did for me to make the original slide. But some of you might relate to that story. But we used to do that, really. At any rate, the nitrate concentration shot up like a rocket to about 60 milligrams per liter of nitrate, which is about equal to the Public Health Service limit for safe drinking water. So all we did was cut down the trees, and the nitrate concentration rose to very high levels. We did not expect that at all. Then the second summer, it rose to even higher levels, more than 80 milligrams per liter, approaching 2 times the level suitable for drinking water. The water looked perfectly clear and drinkable, but it was not. It was polluted by just cutting down the trees. Okay, what was that all about? How did that happen? That was totally unexpected. I'm not a chemist, but I've done an awful lot of chemistry now in my career. At that point, I didn't believe the results, so I did every single kind of chemical analysis I could to verify that the nitrate was real. I even used a Hawk kit. Any of you ever use a Hawk kit? You have. I even used one of those to see. They all gave the same answer, so it was really occurring.
Question?
Yes, where? Yes.
You said that you were expecting the volume of runoff to increase when you cut down the trees, was that in fact the case?
Absolutely, and in summertime it increased about 400%. So it was
the
a big.
volume is higher and the nitrates. even
So the
higher.
concentration, I'm going to get to that. So the concentration times the volume meant that a huge amount of nitrogen was lost downhill. Some of you might have read some of Aldo Leopold's work, Sand County Almanac. If you haven't, I would urge you to read it sometime. It's a wonderful philosophical book about the land ethic. Leopold talks about the loss of materials downhill and escaping from the systems where they are used and are required. And as they move downhill, they ultimately find their burial in the ocean. And so this Odyssey of Adam X that he talks about is that story. Well, that's what this is. We're losing elements downhill. Nitrogen is a critical element for these systems, and with this disturbance, they're moved downhill and no longer available for the plants to regrow. If your garden, if you keep taking away from your garden every year and don't put back anything, after a while it's not going to produce good carrots anymore because the productivity is gone. Same process, exactly. Okay, so what we ended up, and I'm making all this story very short, what we found out was that clear cutting down all the trees, increased the transpiration, increased the transpiration. Without the trees, we had a warmer and moister soil, we had more stream water flowing. At the same time, the microbial controls between the plants and the microbes were released because we had eliminated the trees. So we had more nutrients, as I've described, more dissolved substances, more in solution. The export was hugely increased, and the system was being degraded, seriously degraded, as a result of that disturbance. Well, nobody knew that at the time. So this created a big set of questions and reactions. A lot of my work seemed to cause reactions. The forest people said, well, this isn't the normal harvest, and so it's an exaggerated result. The people that were working in Vietnam said, well, this is just like what we're doing in Vietnam when we add defoliants 2,4,5-T and we're killing the mangroves, and this is exactly what, it wasn't anything like what was going on in Vietnam, and on and on and on. So this was a new finding that required us to spend a lot of effort trying to understand what was happening. So, and I'm not going to talk about this today, I don't have time. So what we did was we did a variety of experiments that were related to forest harvest. We also did one where we tried to harvest in a way that would allow us to remove the timber, but to maintain the system's integrity without losing large amounts of nutrients. And so we did a number of experiments in our vessels on the desk to try to find out how this whole thing worked and how it could be improved in terms of the impact. We also, and I'm going to talk about this either, I'm trying to give you an overview of the kinds of things we did. We did a whole series of stream manipulations, manipulating, experimenting with streams to try to find out how they function. We added things, we took away things, we covered a whole stream over and made it shady. We took all the vegetation away from streams so that it was sunny, and on, all kinds of things we did to try to understand how they were working. Here's one where we were adding chemicals to a stream by hand and trying to understand how the stream responded. You do some of that work here on campus with the salt work that you do on campus. So that's what we tried to do to understand how the system is working by doing the experimental manipulation. The system, as I said, is very diverse. The Neotropical birds, these are all birds that breed at Hubbard Brook. They live in the wintertime in warmer climates like in the Caribbean or Mexico or somewhere. So they fly every year from those warmer climes to Hubbard Brook. One bird was tagged and found to come back and forth that long trip 9 different years. So it was recorded 9 years, which is a remarkable set of movements for a bird. So these are some of the neotropical birds that are studied, the variety of plants that are studied. We put all of that information together in terms of the food webs and how they're analyzed. So we have different sources of food, fruits, foliage, woody tissue, all this kind of stuff that we have to try to understand, and found out that most of it goes to this decaying part of the food web. Relatively small of it, part of it goes to all these things that you're familiar with in a forest, like the birds and the chipmunks and the mice and the deer and the whatevers. Most of it goes this way. Sorry. The birds and the salamanders have been looked at for a long time, and what's been found is that the bird populations have declined by about 70% since the studies were started in 1969. So the decline has been about 70%. Salamanders have declined about 74%. So there's been a big decline in their numbers, but not all. The American redstart has declined. The American flycatcher has disappeared from the forest, but the red-eyed vireo has stayed about the same, and the black-throated green warblers actually increased. So that raises more complexity. Why are some species declining, even disappearing? Others are staying the same or increasing, and that turns out to be because of, well, first of all, it was suspected it might be because of what is happening to their habitat in the warmer climates like the Caribbean. But no, it turns out that it's a result of how the forest has changed with time, that the forest is changing with time, a process we call succession, and that changes has made for different habitats, different breeding and different competition systems between these birds and what they eat, and so that affects different species very differently, and we have to try to understand all that. So the mortality, it turns out, for birds, and the same would be probably true here in Maryland in rough form, mostly occurs when they're having their spring migration back from the wintering population here, or their fall migration down to the warmer climates. It turns out that about 25 to 30% of the mortality occurs on these long flights, each one of them. So maybe 60% of the mortality for the year, oh shoot, sorry, my thumb's too big. 60% of the mortality is due to these migration periods, whereas when they're in residence at Hubbard Brook in the summertime, maybe the total mortality is only 1% or so. So maybe that surprises you, maybe it doesn't, but this is the first time, as far as we know, that that's ever been quantified in a reliable way. So we now know, we suspected it, but we now know with numbers that that's actually occurring. The mortality that occurs, it comes from all kinds of sources. This is a chipmunk taking chicks out of a nest at Hubbard Brook. Some of you may love chipmunks. You think they're cute, nice little critters. They're not. Particularly if you're a bird, they're not nice at all. They're a major predator. And so these are, with cameras, we can see all kinds of predators, and chipmunks are one of the main ones. Okay, so those of you that were there yesterday saw this, but I'm showing it again and a few others to make the point that we discovered acid rain at Hubbard Brook. That first sample that we collected was about 100 times more acidic than we expected that it should be, but we didn't know why, and so that led to a variety of studies we had to do to try to understand and know what to do about that. The questions that we raised were, how is acid rain formed? How long has it been acid, and what was the pre-industrial revolution number? How widespread is the acidity? Is it unique to Hubbard Brook or more widespread? And then what is the source area for the acid rain? We had to answer all those questions in order to move ahead with our understanding of acid rain. So acid rain is formed when the combustion of fossil fuels produces sulfur and nitrogen oxides to the atmosphere, and then sulfuric and nitric acid get formed and come back to the land surface in both wet and dry deposition. The trajectory, this is not yesterday, this is new. The trajectory, if you plot the sum of base cations on the vertical axis against the sum of sulfate and nitrate, which are the acid-forming anions, goes something like, something like this. From looking at remote areas, we're able to establish what the pre-industrial revolution number might have been. We think about pH of 5.1, and that brings this relationship right there. And then moving up to the very first sample that we collected in 1963 and then 64. So this is the period of acidification that we caught at Hubbard Brook. It was occurring for a long time, probably since about 1950 or the mid-1950s. We don't know for sure, but we think that's about right. But this period here is when acidification was occurring. 64, 65, 66, 67, 68, 69. Isn't that remarkable how it marches up that line just right? You don't need any statistics for that. And then the hysteresis turnaround, and 70, 71, and right on back down, headed toward that pre-industrial revolution value. So that's the acidification recovery curve for Hubbard Brook as a result of acid rain and acid rain impact. We had to answer the question of, was this unique to Hubbard Brook? So setting up stations in the Finger Lakes region of New York, I could determine that the chemistry there was almost the same as what it had been at Hubbard Brook, and that was the first clue that we had that it was a regional problem, not just something occurring at Hubbard Brook. As you know, the wind blows from west to east, bringing pollutants from the Midwest to the east. 70% of the sulfur dioxide is produced by the electrical generating facilities in the Midwestern United States. Huge amounts. Ohio, Indiana, Kentucky, Illinois, that's where most of the coal and oil, particularly coal, high sulfur coal, is burned, producing the pollutants that are then carried to the east. So the problem really was these pollutants from the Midwest were being dumped on Maryland, but being dumped on New England and southeastern Canada, and nobody liked having that garbage dumped in their backyard. And that's what it was viewed as. Your pollutants are being dumped in my backyard. I don't like it at all. So the relationship between the relationship between the sulfur concentration in rain and the emissions looked like this, where we could see eventually with the long-term data this relationship being quite strong, showing that there was a major relationship between what was emitted and what we were collecting at Hubbard Brook. So I talked about this a little bit yesterday, the acid rain wars of the 1980s, and this large source in Canada, the copper-nickel smelter at Sudbury, Ontario. And I said yesterday that the emissions from this stack, when it was in full operation, was more than the sum of all the active volcanic emissions around the world. So it was a huge amount of sulfur emitted from this stack. So this led to the negotiations between the US and Canada about transboundary transport of pollutants. The Carter administration was developing a memorandum of intent, which President Reagan then terminated when he was elected after Carter was defeated. And this cartoon I showed you yesterday that related to Brian Mulroney in Canada, the Prime Minister, and President Reagan exchanging this cartoon satire about, sir, all Canadians pray for a solution to the acid rain problem. Well, gosh, Brian, you tell them that Nancy and I join them in their prayers, but we aren't going to do anything about it. Sorry. And then I led a small delegation to brief President Reagan in 1983 in September. I didn't have time yesterday to say much about this photograph, but there's Ruckels's house. Here's Keyworth, who was the science advisor for Reagan at that time. Here is Reagan with his back and making a presentation about acid rain, what it was, what the damage was causing, and what we should do about it. The decision was made not to do anything about it, but to study it. And so that was what was done through a program called NAPAP, N-A-P-A-P, National Acid Precipitation Assessment Program. We spent $570 million as a country to study acid rain for a period of 10 years. The change in emissions of sulfur dioxide and nitrogen oxides to the atmosphere because of federal regulation has been a huge success story. The data from Hubbard Brook, 50 years, show that the acidity is now about 80% less than it was when we started to make our measurements. So we're here. Still not back to where it should be if it was an unpolluted condition, but it's not. So, but this is a major success story in terms of environmental regulation, which we're in the process of trying to turn around right now. The inputs of acid, sulfuric and nitric acid to the system causes an exchange of calcium and magnesium as the hydrogen ion exchanges with them in the soil system, producing more calcium and magnesium in the soil water. Aluminum is dissolved and released, and that all can then leave the system in stream water. The forest at Hubbard Brook has stopped growing. The forest was cut heavily in about 1920, that period, and was expected to regrow to a condition. Our models suggested that the regrowth should end up way up here somewhere. But by the time we got to 1982, that didn't happen. It flattened off, and the growth stopped and now has declined, so that statistically there is less growth than there had been. So the system is now degrading. The system is no longer sequestering carbon from the atmosphere because the soil carbon, by our measurements, is roughly constant. So it's no longer sequestering carbon from the atmosphere, it is emitting carbon to the atmosphere. That's a major, major factor, if true. But that's what our data show. I showed this yesterday, but I didn't really have time to talk about it. So different species respond differently to acid rain. Red spruce, a conifer, The calcium is leached from the needles by direct input of acidity on the needles, actually leaches the calcium out of the needles. This is work done by the University of Vermont scientists. That makes those needles much less resistant to cold. So when it's a really cold winter, those needles die, and if enough of them die, the whole tree dies. So throughout New England, this is a common phenomenon. There's also some effects through the soils, but for something like a sugar maple, and I haven't had a chance to look on your campus, but I'm sure you must have sugar maple decline. These are the classic symptoms of sugar maple decline, where the top middle part of the tree dies first, usually turns color first, and then after about 5 years, the whole tree dies. At the University of Connecticut, where I'm currently residing, all over the campus, these big, beautiful old sugar maples are all in sugar maple decline, all of them. I've urged them to try to add calcium, because one of the reasons for sugar maple decline is that there's a large amount of calcium and magnesium that has been leached from the soil. They're calcium-loving trees, but the trees have already gone past the limit where recovery is going to happen, so they're just being taken down. They're dying. It's really, if you like trees, which I do, it's really sad. Aluminum, which is leached, is very toxic to tree roots, toxic to aquatic organisms as well. So that both factors, aluminum toxicity and decline of calcium and magnesium, is occurring to cause these major effects. So we did the experiment in which we calculated all the calcium that had been depleted from our system in the previous 50 years, and that was about 500 kilograms of calcium per hectare during the period 1940 to 1995, and we added it all back experimentally. Again, as I said, Hubbard Brook is an experimental forest, so we do these large-scale experiments to try to understand. Experimentation is very powerful in science, as you know. So we do these experiments to try to understand what's going on. So we added back all that calcium and some more in a mineral called wollastonite. Wollastonite was used because it's a more gentle acid neutralizing component than, say, calcite or lime. The problem with wollastonite is it has another nutrient in it, silicon. But we added wollastonite, had it ground, had it mined and then ground, and then pelletized so that we could handle it, and then by helicopter added it back with the helicopter flying very careful grids and people on the ground collecting wollastonite pellets that fell so we could quantify exactly what fell and where, and that was done. And the effect of that was to produce a recovery in the forest. So the sugar maples then recovered and are recovering, and these are data recently published that show in the red line here the recovery of the area treated with wollastonite and the reference watershed. We can't really call these controls because ecosystems are too complicated to have a strict control. So we don't use control, we use the term reference. But you have to have a reference because if the climate changes or something else changes, you have to have a reference to know what effect your experiment had versus what some other factors may have had. So it isn't a strict control, but it's like a control. Very important point in these large-scale systems. And this is a red spruce injury after a very cold winter, but with wollastonite treatment, the trees were protected and are now being protected by the added calcium that we've added to the system. As I mentioned, one of the things that we've discovered, really, I think that's an appropriate term, from this work is that calcium is a limiting nutrient for this system. We didn't know that, nor did anybody else. We thought primarily nitrogen, maybe phosphorus, but primarily nitrogen, but it's calcium. in this degraded state. That was this slide. It's why I repeated all those slides about acid rain. This is probably the most remarkable slide I have to show you. I did not show it yesterday. What this slide shows is that the precipitation chemistry and the stream water chemistry at Hubbard Brook are now on robust trajectories toward distilled water. You can't have a biological system living in distilled water. So theoretically, it's not possible that these robust trajectories can go all the way to the baseline here, where these trajectories show the R squares on these lines, whether linear or quadratic, are more than 0.85. So again, you really don't need statistics. So two things. Are they really going to go to very low values? We said, well, I don't think we can justify extrapolating them to 0, even though that's what they show. So we picked theoretical stop points here, 5 microsiemens, 3 microsiemens per centimeter, and said that's where they must be going to taper off. And they're going to do it right now. So you guys are going to be around to see it. I hope I am too. But it's happening right now. This is a question that we're answering right now. It's one of the reasons I want to know every year what the latest data are. What's happening? I don't think it's going to go there, but that's sure where it's headed. That's one point. The other point is why? What is happening at Hubbard Brook? The precip chemistry almost certainly is on this trajectory because of our success with environmental regulation. We have cleaned up the pollutants to a point, not as good as we need to and as we should, but to a point that the system is now becoming very dilute as a result of that success. What about stream water? Well, we've leached out the calcium and magnesium from the soils by this constant rain, sorry about the pun, of acid rain into the system, so that there's now very little that can be weathered and leached into the stream water. We've cleaned up the atmospheric pollution of particulate calcium and magnesium. It's what the 1970 Clean Air Act did. It's what China is faced with right now, big time, to the point that we're overshooting. What's good? I'm laughing. I don't know whether to laugh or what. This is a success story. So I should be cheering, but not if it goes to distilled water. That would be a catastrophe. So what's going to happen? I don't know, but I'm sure anxious to find out. You all with me on this? That's why I had to repeat all those acid rain slides. For those of you that didn't go, and even those of you that did go yesterday, to show you this slide. I have one more, but this one primarily. Any questions about this before I move on and finish? Yeah.
Isn't that distilled water going to be very acidic and just hold more calories?
Actually, the distilled water is probably not going to be that acidic. It's probably going to be around 6-ish, I would guess. Probably. But again, I don't know. So we published this dilutification, a new term for you. There's eutrophication, and there's acidification, and there's salinization. This is dilutification. Nobody else has reported this. Nobody else has these long-term data. Another advantage of having the really long-term look, the long-term perspective. So again, I don't know what this means. I'm really curious to know what this means. And yeah, I'm getting older, but I'm really curious to know what this means. And I'm going to stick around long enough to find out what this means, okay?
So it was 2 years on from 2016. So
Yeah, but yeah, and well, I may come back to this in a moment, but if we roll back these air pollution controls, then, you know, then these graphs may well not only flatten off, they could go back up. And that also has impact because the system is so, it's lost so much buffering capacity that that additional acidity could be very bad. So yeah. Okay, one last moment, any questions? Okay, moving on. So it takes a long time. In this case, it took 27 years and 3 presidents and 1 pope. A pope issued an encyclical after we had a week-long meeting at the Vatican. And I looked into some other long-term environmental problems like lead, like DDT, like eutrophication, and every one of them took between 20 and 30 years between discovery and some kind of action, some kind of environmental action. So from that look, it appears that it takes us that long to decide that, well, the vested interest really, it's all about vested interests and money. And if we're really going to protect the environment, this is what we're going to have to do. And it may go through, in our case, several administrations before they get the nerve and the political will and the public opinion to do it. But it takes that long time. For those of you that are in your early 20s, think of the next big environmental problem, which is coming down the pike, and they are. And it's going to be 50, you're going to be 50 by the time they resolve it. Can you wait that long? Yeah, I don't know. The costs are very clear. Even with however you want to manipulate the economic analysis, the cost-benefit ratio still is strongly skewed toward we do get our money's worth, we protect our health. One of the biggest costs of acid rain is not us breathing in acid rain. I had letters written to me from people saying, you know, I was out in the rain and I'm afraid it's going to dissolve my hair. No, it's the particles, the sulfur dioxide particles, 2.5 micrometers in size that you get lodged in your lungs that causes problems. That's where the big costs come in. We've been, with the long-term records, able to look at some of the changes in climate. So the mean annual air temperature is increased, the winter temperature is increased, the summer temperature has increased, the soil temperature has increased. This is showing for August. The amount of precipitation is getting wetter at Hubbard Brook. The amount of snow is decreasing. We're getting a lot more rain on snow events. I don't have time to talk about that, but it's a very significant feature ecologically. The amount of ice cover on the lake at the bottom of the valley is decreasing. The ice cover on a lake is a very good indicator of the total heat balance for a lake. There's a larger version of that, and 2 things have happened. It is the amount of the length of ice cover is shortening, and the variation year to year is increasing. So variation here, sure, but now it's going like this. So those 2 things are clearly happening, and ice cover on lakes is one of the really good ways to look at local climate change effects. So climate change indicators are all over the place at Hubbard Brook. Buds break on the trees earlier. color comes on the trees in the fall earlier, the growing season is longer. All of this has been documented in our long-term records. So, what are the major lessons we've learned after 55 years of looking carefully and doggedly at the system? There's been major effects on the biogeochemical flux and cycling. We've learned that stream ecosystems are an integral part of all of this. It's not just the trees, or it's not just the soil, or it's not just the lake, it's not just the rain or snow, it's all of this, and streams are an integral part of that, and a good indicator, like the physician, of whether your blood chemistry and urine chemistry is telling you something is going wrong. Long-term changes in biodiversity that I described with birds and salamanders, the discovery of acid rain, the climate change, and where do we go for the future. In 25 years, we're now 55, in 25 years we had a celebration. We celebrated the fact that we made 25 years. And these were two of my collaborators. There were four of us that started Hubbard Brook. Unfortunately, three of them are deceased. I'm the only one left. But we were having fun, and I'm still having fun. And I said, I think, to you guys, the students that I've talked with, that you really have to have fun. This is hard work, and if you don't have any fun, you're not going to do a good job. So I think fun is a critical part of doing research. I cannot ever remember in my adult professional life waking up in the morning dreading to go to work. I don't consider it work. I love to go every morning. And I think that's what science is. Look at the wonderful opportunity we have to find out stuff. If you are inquisitive, you can find out stuff. And sometimes you can be lucky to find out stuff that really matters. You can travel. You guys were telling me about all the places you travel to. Good grief. And I've been able to travel. Don't miss having fun. It's a wonderful occupation. So I'm going to leave it with that slide. The value of the long-term data gives us insights we don't have any other ways. Some of you look at long-term data by looking at sediments or cores or whatever. I do it in that way too, but I also have this long record of empirical measurements that I've made. We need our public policy based on science, I would argue, maybe never before like we are right now. The importance of putting it all together. Piecemeal doesn't work. Silos don't work. We need it all together to understand what's really going on. I talked to a few of you about ethics. I've taught a mini course in scientific ethics for a long time, 20 plus years. Don't cheat. Ethics are so important. You need a lot of pressures to get ahead, a lot of pressures to get tenure, to get a job, to get an award, to get whatever, but don't cheat. It's too good a job. And the importance to have, as I say here, visionary and intelligent leadership. So we have to work really hard. I think I'm preaching now. I'm not apologizing for it. We have to work really hard to educate our leaders about what science is, what we do, why it's so important, and what it means. And we have to learn to talk to them without jargon. We have to learn to talk to them so that they can understand why all that money the public gives us is worthwhile. Thank you.
I don't know if you guys are like me. I thought it was just half an hour, but it's fast forward.
Oh, I'm sorry.
No, no worries, that was excellent. So, questions? Yes, sir.
I was just wondering, so we've had this big curve in all of these particulates and the sulfur. Did that have a noticeable or calculable heating effect on the climate because we got rid of all of this sulfur emission?
I don't know. I don't know. I know that it has been proposed to put sulfur compounds in the stratosphere to try to combat climate change. I don't know if there's been any effect of this shorter term, smaller regulatory effect. I don't know the answer to that. Sorry. I could guess, but I don't know the answer to it. Sorry?
I think there's some evidence that the answer is yes since like the 50s. Yeah.
Did you hear what he said? Say it again louder, please.
I think there's some evidence, yes, that we masked some of the warming that we would have had from greenhouse gas concentrations by aerosols, which have a cooling effect. So you remove the aerosols, you allow the full expression of the greenhouse gas.
And there's another aspect that's different than that, but sulfur is a nutrient. It's a critical biological nutrient. And back in the very early days of the acid rain wars, there was a lot of arguments that we can't take all this sulfur out of the atmosphere. We need it for plants. And now that argument is appearing again. And when my dilutification graph that I showed you, excuse me, it's there as well. So again, we can't deal with these things piecemeal. We have to think about them in a comprehensive, collective system sort of way if we're going to make any progress in moving ahead, in my opinion. Thank you for answering the question. There was another hand I saw. Yes.
So, as someone who studied or experimented in open forests, what do you think about biospheres, like the one in Arizona and France that do experiments but in a more closed system?
Oh, I have complicated views about that. Yeah, I was actually involved in that early on in Arizona. Let me answer it this way. I'm weaseling out of answering it, but I prefer the natural system experiments if it can be done. They're both expensive to do. At Hubbard Brook, we've now essentially used up all the watersheds that are available for experimentation, so we can't do any more without an elaborate building effort to make more watershed ecosystems. I think those two different approaches are useful for two different things. I don't think my watershed ecosystems are very useful for space travel, whereas the Arizona spheres are maybe more useful for understanding about space. You guys probably know a lot more about that than I do, but about space considerations. But I like my way. I didn't say it was better, I just said I like my way. Yeah.
The bird declines that you documented, I noticed that the flycatchers disappeared, and I wondered if you also tracked the decline in insect populations, particularly flying insects, which is experienced a worldwide decline.
Yes, our data aren't as good, but yes, the answer is yes. We have tracked it, not as well, and appears that there is some decline there as well. But our data aren't nearly. One of the things about Hubbard Brook is we never either had the philosophy or the money to go out and say, oh, we need somebody to come in and study the arthropods in the system. The way people come into Hubbard Brook is because they want to. They, I want to study something. Well, okay, if it doesn't, directly conflict with what somebody else is doing, sure, come work at Hubbard Brook. For long-term analyses, it'd be much better to have maybe, I don't know, but maybe to have this big vision. So we need to hire somebody to do this and hire somebody to do this and we just didn't do it. We didn't do it that way.
I was going with these wonderful observations you did with Hubbard Brook, is any other site around the world in a
Yes.
different type of climate or influences
Yeah,
that are comparable?
There are,
So that-
there are a series of experimental watersheds in the United States. Fresno in Pennsylvania is an example. Coweta in North Carolina is an example, Coweta Hydrologic Laboratory. Andrews Forest in Oregon, there's a number of them. Those are examples. None of them quite went the way we did, where we experimentally manipulated whole systems and also maintained long-term monitoring records, as Sujay, I think, said when he introduced me. I think these are the longest comprehensive records for precip chemistry and stream water chemistry in the world. I think that might be the case. Those other stations didn't go that way. They didn't do that, but they do exist.
Yeah, so I was wondering if these are existing, the synthesis of this must show up something like in common or also contrasting.
They're very different. They're very different systems. And one, I don't think anybody's really carefully looked at that in that way, in that systematic way, but they're very different systems. And so Andrews in Oregon doesn't have acid rain. It's near the coast. It's a Corvallis, near the coast. So most of their atmospheric inputs come right off the Pacific Ocean and are relatively clean. Ours come from the Midwest and are very dirty. So they're different.
So following up what Linda asked, so you showed that the diagram of wind directions, so it shows that sort of New England is sort of a trash can of North America. So like your results might be over exaggerated than the very
For the whole country.
right.
Absolutely. So I would say Hubbard Brook is only one place, and that's why we pushed so hard back in 1975, 76, 77, 78, got it happen, to establish a network all over the U.S. I think there's 253 stations now all over the continental United States measuring precipitation chemistry, and that's why we did that, because Hubbard Brook is just one place. But Hubbard Brook, I think, is the canary in the coal mine for the northeastern United States, the southeastern part of Canada, maybe even as far down as here, the Atlantic coast states. But yeah, it's only one station. I couldn't do more than one station.
My understanding is that those are Maine, New Hampshire, and those are big agricultural states. And
No, they're not.
Well, but my understanding is they do artificial rainfalls, and they spatter the aluminum to induce rain.
I'm not sure I understood that question.
So does that affect yours?
Again, please?
Oh, so they induce artificial rain. Yeah.
He's talking about irrigation, and it's not.
Oh, there's relatively very little irrigation in
Yeah.
even Massachusetts and Connecticut. There's some, but then as you move up to Vermont, New Hampshire, and Maine, there's very little. There's some, but very little irrigation. Very little. Yeah. Sorry, I didn't understand your question.
But one of the things that is very different about Hubbard Brook and a lot of the other areas that surround it is it does have very, very thin soils
Yes.
and granitic
Yes.
bedrock. And
Yes.
so when you compare the solute concentration in the stream chemistry with, let's say, what we collect from the USGS National Water Quality Assessment Program, it does have some of the lowest
Yes.
of stream solute concentrations.
Which for our studies were wonderful, because arguably we would never have seen the impact in the stream water if it had been highly buffered. But because it wasn't highly buffered, and in fact the location of those weirs, we published a paper on this, the location of those weirs in the whole valley are like here, and it's almost like a titration curve. They're right on that inflection point in the titration curve. If they had been located here or there, We might have seen it, but it might have taken us 20 years. But where they were located, we were fortunate we saw it immediately. So for our work, it was ideal. But it's only one site. Absolutely legitimate point. Yeah.
The student first.
So you kind of alluded to my question in your last answer, but I wanted to ask about potential buffering, about how New England is mostly metamorphic bedrock, and how if you have a similar process that's happening in the Midwest, where there's a lot of carbonates as the bedrock.
And there is in Vermont. Vermont, right next door to us, there's a lot of limestone.
Oh, there is. Okay. So does a limestone bedrock, does that affect the deposition?
Absolutely. No, not the deposition. It affects the stream water chemistry, big time. And so the Hubbard Brook Valley is granitic, and we have leached away more than 50% of the exchangeable calcium and magnesium in our soil system. So more than half of it has been leached away. If we were down the road in Vermont where they have a big limestone outcrop, they don't see that at all. So yes, again, I can't say it enough times, you gotta look at the system and all the parts of the system, yes. But that makes it also really hard, doesn't it? I mean, there's only one Hubbard Brook. Be nice if there were 100 of them, but there's never going to be. And so I think we have to take from a place like Hubbard Brook and use it to take those questions out and address them in different environments and different locations, and then try to put it all together. I think that's what we have to do.
Is it a short?
I'm sorry.
I'm curious what your prognosis is for the continuation of the monitoring at Hubbard Brook.
We're now part of the LTER network, and we have just gotten 6 more years of funding from NSF. Beyond that, who knows? Who knows? From very much like David Keeling, who I knew very well, they tried to shut him down 6 different times. And if you go on their website, you'll see where they tried to shut him down. The question always is, haven't you studied this long enough? Don't you know everything that's going to happen? Why do you need to continue? Why do we need to keep giving you money? So the only way Hubbard Brook was able to persist was by asking really compelling questions that the reviewers said, okay. We were fortunate, I don't like the word lucky, we were fortunate to discover acid rain, so that generated a whole series of questions. We were fortunate to get that surprising result from the deforestation. That gave us a whole series of questions that we could pursue, but it had to be driven by questions.
Okay, this thing is this line.